where you have like starting the stream.
अच्छा हाँ
Yeah, it is now live. We are
Instagram.
live now.
Okay, okay. Just Rishabh is there. Okay, okay, fine. Thanks.
We are live on YouTube. You all can start.
Good morning, ma'am. Hello, everyone. Welcome to our webinar on craniofacial development. Before we kickstart things, I want to give a quick introduction to STEMONEF Community Club. We are all about bringing together folks who are passionate about science, tech, engineering, and maths. So, whether you are a student just starting out or a seasoned professional, our community is a place to be if you are into STEM. We have got all sorts of cool stuff like projects and webinars and articles, everything. We are part of a bigger STEMNF network, and our goal is simple like learn, share, and push boundaries of what's possible in everything around STEM. Thanks for being here with us today. We're stoked to dive into the world of craniofacial development with you all. All right, let's go. Is Kajal here?
Yes, I am. I'm sorry, but I wouldn't be able to keep my camera on, like due to some problems. Is it okay?
Yeah, fine.
Okay, so let me continue. Hello, everyone. It is a privilege to introduce Dr. Shruti Tufane, a distinguished clinician scientist dedicated to understanding craniofacial development and disorders, conditions that arises from abnormalities in the growth of the head and face. Dr. Shruti's academic achievements are remarkable. She earned her BDS from GDC Mumbai, followed by a master's degree from the University of Dundee and a PhD from the University of British Columbia, where her research focused on uncovering the genetic, cellular, and molecular mechanisms that govern the craniofacial formation. Currently, Dr. Shruti is a postdoctoral associate at the University of Minnesota, where her work centers on neural crest cells, the foundational elements of craniofacial development. In addition to her research, Dr. Shruti has presented her findings at global conferences, published influential studies, and contributes actively to STEM education as the head of the biology team at STEMONEF Community. We are honored to have Dr. Shruti here today to share her expertise on craniofacial development and disorders. Please join me in welcoming her to the session. Ma'am, please continue. We are really happy to have you today with us.
Thanks for the kind introduction, Kajal. That was quite overwhelming. Awesome. So what are we doing now? Is somebody sharing the presentation?
Okay, ma'am. Now you can share it. I'll stop sharing my screen, and now you can just share PPT.
Okay, just give me a minute. Do I share the entire screen or because I don't see my PowerPoint here?
The entire screen. Yes. Then you can open it.
Oh my God. Okay. I'm sorry, I'm not so used to Google Meets, so just bear with me if I'm just, doing some random stuff. Okay, so do you see my presentation?
Yes, ma'am.
Okay, so let's see. Is there a way where I can see the participants as well, or I just see my own screen?
I think you can minimize the Chrome tab, then then it will appear. You can open the Chrome tab and then minimize it.
Okay, let's see.
Like, click the minimize button, then it will appear.
Okay, I don't know how to do that. Sorry.
Just open it, ma'am. The Chrome tab.
The Chrome, okay.
Yeah.
And
then click the minimize button there.
this one?
Yeah. It will appear then.
No, I mean, it's fine. That's okay.
Oh, it is not appear. Okay, it's okay.
That's
it
okay.
Does. Yeah.
Okay, so basically what I'm going to be talking today is about development of organisms, specifically focusing on human development. I'm not specifically going to concentrate on development of the craniofacial region because I thought it would be better to have an overall view of how we develop as organisms. So this talk is a pretty basic walkthrough through human embryonic development, starting from a single cell to how we look like we do. So Kajal has already introduced me, so I'm not going to waste a lot of time, but I have to say that the journey from being a clinician to a scientist has been quite rewarding. Being a dentist, I understand the clinical aspect of diseases and how many emotional and other aspects are also involved in a certain disease manifestation along with the pathological features. But now being a researcher, I get to address it by understanding the pathogenesis of diseases. So it's a quite a rewarding career, but it has its stakes. As you all know, it's a very, very dedicated profession that if you are really passionate about development or research, you can definitely be in one. So yeah, I have a bachelor's in dental surgery, a master's in oral biology, and a PhD in cell and developmental biology. And currently, I am a postdoctoral researcher at UMN in Minnesota. So I would be happy to discuss my career path and how I went from being a dentist to a scientist later on after the talk, if anybody is interested. So moving on to the actual presentation, focusing on understanding why we care about developmental biology. So we care about developmental biology is because it is the basis of our existence. It is a necessary thing that we need to understand how we develop from a single cell or a multicellular stage to a full grown human. So another aspect of developmental biology is that if something goes wrong during the process of development, there are severe disease manifestations, as you can see here. So you can see craniofacial developmental defects, like severe developmental defects, diseases in which the two siblings are like conjoined twins kind of thing. You can also see, so all of these on the left side are something to do mostly with craniofacial development. This is a character that that Mr. Amitabh Bachchan played. So this is progeria, where the entire body is affected, and it's like the body ages faster than it actually should. On the right hand side, you see some dwarfisms where the the person is extremely short. You see extra limbs. So this kid has like 6 hind limbs, and this little baby does not have any limbs. So anything going wrong during development can lead to mild to severe manifestations, as you can see here. And there are multiple diseases that start at very early stages during development. So these are some of the things that I'm going to be covering in the talk today. So starting with embryonic development. So embryonic development is, as we know, a 9-month period, but there are the first 8 weeks of development that are quite crucial. So these are called as the Carnegie stages of development that go from week 1 to week 8 and are divided into 23 stages. So as you can see here, you start as a pinpoint and eventually develop into a full-grown organism. So in the first part of the presentation, I'm going to be covering these Carnegie stages of development and trying to understand what develops when. So moving on to the first week, moving on to just an overview of the Carnegie stages of development. So it starts on day 1, and you are taken through a journey until about 60 days of development. So as you can see here on day 1, it's just like a point. And then on stage 23, you see a full-grown baby with most of the features, including the craniofacial, the limbs, the internal organs are completely developed. So this Carnegie system was designed at a University of Washington, and there's multiple universities that contribute to this in understanding how humans develop. So Carnegie system was developed in like about 40-50 years ago, when it was okay to have human specimens. It was okay to use human specimens for research. And that is when they collected all of these embryos are real human embryos, where they collected these embryos and looked at the different features of development. So I'm going to be talking about the first 8 weeks of development and what develops when. So beginning on the day 1, basically, we all start with something that we know is fertilization. That is, the egg and the sperm unites. So basically, in the human ovaries, there's ovulation taking place. And on the left-hand side, you see this video where you see the sperm entering into the egg and then formation of the pronuclei. So basically, this process here on the left in the video is something called as fertilization, which is counted as day 0. After fertilization, there is a zygote stage, and then division of this into from a single cell, that is in day 1, into a 2-cell stage, into a 4-cell, 8-cell stage. And this is eventually, basically from 2-cell to 8-cell is called as the morula stage. And after this, it is, again, the 8-cell divide. So everything is dividing very, very fast. So you see from day 1 to day 8 or 9, what happens is the embryo is dividing extremely fast, and eventually what it will do is the embryo will eventually implant into into the uterus of the female. So this happens during the week 2, which is again the stage 5 or 6 of the Carnegie stage of development. So basically, you start on day 0. By day 9, the implantation has already occurred. So this is particularly during the days when the female is not even aware that all of these things are happening, right? Moving on to the week 3. Week 3 is where an extremely important event is happening. So basically, in the last slide, you saw the implantation of blastocyst. So blastocyst is basically just like a disc. So you move the disc in any direction, it will look the same, right? After this, in the week 3, this important event called gastrulation is occurring, basically meaning formation of the gut. So during the week 3, what happens is the bilaminar embryo, which is also called as the embryonic disc, it transforms into a trilaminar embryo. So basically, there is a formation of different layers. The first layer, that is the outermost layer, which will eventually form your skin, is called as the endoderm. The middle layer is called as the mesoderm. And the innermost layer, which lines our inner cavities, is called as the ectoderm. So on here on the left, you see formation of primitive node and formation of primitive streak, which is an extremely important event in human development. The reason being that this decides what is left and what is right, and what is your head and what is your tail. So here is going to be your head, here is going to be the tail, and this is what connects the embryo to the mom. So basically, now we are at week 3, that is gestational week 5, because it is counted from the last period of the female. So at gestational week 5, this extremely important event called gastrulation is occurring. If something like this does not occur, if gastrulation fails to occur, then the development is not going to proceed. Moving on to week 4. So week 4 is where the actual development is occurring. So now the embryo looks like an embryo, something that you can tell is a head, something that you can tell is a tail. So in this, on the right side, you see the head of an embryo. This is the body, and this is the tail. I mean, we don't really have a tail, but this is the tail. Eventually will disappear. Here you can see the limbs, which is going to form your hands, and this is the heart. So basically, what is happening at this stage is the formation of different organs is occurring. Especially the heart is the first organ that will form. So basically, the neural tube, which we saw before, that is the primitive streak, it eventually starts closing. In the head region, you see formation of different pharyngeal arches, which may be a very technical term for you guys, but pharyngeal arches is something which will form your upper jaw, it will form your lower jaw, and it will form the inside of your neck. At this point, you also see, it may not be very obvious, but this is where your eye is going to form. And at this point, the ears also start to appear, and the oral cavity, which is somewhere over here, it will start to appear. The limb birds are starting to form. As I said, the heart is starting to form, and at this stage, the liver development also begins. And at this stage, the embryo starts to feel the sense of smell. So the nasal placodes, which are going to form somewhere here, they also start to appear. So basically, the face development commences at week 4 and proceeds until week 8, when by week 8 the entire face is completely developed. At this stage, the skin, which, as I said, the outer layer of the embryonic disc, which is the ectoderm, which also starts to form. So things are starting to happen at week 4. So week 4 is very, very important. So basically, this is a cartoon or a video animation of week 4, where you see this is basically the neural tube. The neural tube is closing. This is the head. This is the tail. This is the heart. So basically, the embryo, from being a disc to a trilaminar embryo to starting to look like an embryo, it starts to curve a little bit at week 4. Moving on to week 5. Week 5, gestational week 7. At this point, the embryo is about 9 millimeters in size. So as you see now, the embryo is quite curved. So it's trying to accommodate itself inside. At this point, a lot of features are appearing. So beginning from the brain starts to develop. So this is something that will form your forebrain. This is the midbrain, and this is the hindbrain. So all of this region is going to form the brain. This is the face of the embryo. This is where your eyes are going to form. This is the nasal placoid. So this is going to form your nose. These are the pharyngeal arches. So first, second, and third pharyngeal arches is forming here. So this is going to form your upper jaw, your lower jaw, and the pharynx. This little pit here is the otic placoid, which is going to form the ear. Moving on to the body, this is where the heart is forming. This is the upper limb, which will form the hands. And this is the lower limb, which will form the legs. So basically at this point, most of the identifiable features appear, and most of the features start to get more and more defined. So you can tell what is what. Moving on to week 6. So now we are up to Carnegie stage 17. So we went from being just egg and sperm, fertilization, implantation, formation of bilaminar, trilaminar embryo, to forming an actual embryo which has definable features. And this is what we went through the week 1 to 5. And now at week 6, what is happening is everything is now in full speed. All the internal organs are developing. So if you look from the ectoderm point of view, so the ectoderm, which will form the sensory placoids, are forming the lens pit. So everything is quite on full blast at this point. So moving to the head, you see the lens placoid, the ear, the nasal pits. Everything is quite well defined at this point. The heart again is developing. The face is developing as well, and you see most of the internal organs are developing. So one might say that week 5 and 6 is kind of similar, but the main difference between week 5 and week 6 is that in week 6 most of the internal organs are developing. The embryo looks similar, is a little bigger, is about 14 millimeters in size, but internally most of the organs are beginning to develop. And with this, I move on to week 7. So in week 7, again, the embryo is again getting quite bigger. So it is about 17 millimeters in size. You see all the identifiable features, that is the head, the limbs, the heart, but an important feature that forms to appear here. So from being just buds to getting fingers, this is the main event that is occurring here. So you see digital rays, that is fingers being formed at this stage. So here I just wanted to elaborate a little bit on the face. So because in all of the pictures that you saw until now, the embryo was curved, you're not able to see the face. So what does the face look from the front? So this is how, if you turn the embryo towards you, the embryo is going to look like that. So in here, this is Carnegie week 7, Carnegie stage 18. So the face looks like this. So this is the frontonasal prominence. These terms might be too technical, but I'll try to simplify them a little bit. So the frontonasal prominence eventually will give rise to the midline structures of your face, including the forehead, the bridge of the nose, and something that forms a little bit of your upper lip. The medial nasal prominence and the maxillary prominence eventually fuse with each other. These two prominences fuse with each other and giving rise to your upper jaw. So your upper jaw will form with these three prominent with these two prominences, and then the mandibular prominence, which will form your lower jaw. These are the nasal pits, so these will form your nose. So anything going wrong in these fusions, so basically the medial nasal prominence fusing with the maxillary prominence will give rise to something which is called as a cleft, which I'll discuss in some slides after this. If the fusions in the facial region do not occur for some reason, you'll see craniofacial developmental defects like some of those that I showed before. So at this stage, also what is happening is that there are all these fusion events occurring, but before these facial prominences or the other organs of the body are concentrated or occupied by these crest, which are called as neural crest cells, which is something that I specialize in, which is what I'm studying in my current research. So these cells, the neural crest cells, occupy these prominences and give rise to different elements of the face. Especially in the face, what they contribute is the facial skeleton. So at this stage, you also see that the ossification has begun, because you see the formation of digital rays here. So basically in the entire body, the bone is starting to form. Moving on to the week, to Carnegie stage 19, which is again a week 7. So at this stage, the embryo looks quite like a human embryo, but it's extremely small. It's about 19 millimeters at this stage, but you can tell the features. So the head, the eyes, the oral cavity has formed, the upper jaw has formed, the lower jaw has formed. This is something that is going to form the ear, but it hasn't formed yet. It is just a bit. Then you see the upper limb, the lower limb. You still don't see fingers at this point, but you might be able to appreciate a little bit that the fingers are starting to form. Moving on to the last stage, that is the week 8, that is about 60 days after day 0. The embryo by this stage is about 31 millimeters in size, so it's little, but most of the features have formed. This is where I'm showing you a 3D MRI of embryo which was scanned at week 8. So in this MRI, you will be able to appreciate a few features like the brain has formed. The face is quite definable. You see all the internal organs have formed. The limbs have formed. You also can appreciate the limb bones. So let it come back one more time. Okay, so you see the brain, you see the eyes, you see the ears, you see the face, and you see the limb bones and all the internal organs, and you see the spinal cord. So basically from week 1 to week 8 is the most crucial period of human embryonic development. And the reason why we say this is because anything going wrong at these specific stages can lead to severe developmental defects. And Carnegie stage defines these 8 weeks. So after this, please do not think that after week 8 nothing is developing. After week 8, the embryo continues to develop, and the bone is still ossifying, the embryo is still in a stage where multiple organ systems are developing. So a lot of the part of the developmental process is still occurring after week 8, but these first 8 weeks of development, that is the gestational week 10, is when the major morphological events are occurring. So the reason why we care about these is because anything going wrong, as I said, will lead to developmental defects. So we went through the period of the embryo dividing, from a 2-cell to a morula to a blastocyst implantation, and we looked at formation of a bilaminar and a trilaminar embryo. We went through the main embryonic period, that is the first 8 weeks of gestation. As I said, during the first 8 weeks, the neural tube is forming. The neural tube is eventually closing. If the neural tube does not close, it will lead to neural tube closure defects, which I'll show in the next slide. What happens if the neural tube doesn't close? And if this event doesn't occur, then you might have a baby without a brain. If something goes wrong between the week 3 to week 6, you have multiple heart defects like auricular septal defect, ventricular septal defect. At this point, the heart is less sensitive. So from week 6 to week 8, the heart is a little less sensitive to developmental defects. But the first 3 to 6.5 weeks are quite important for heart development. For the upper limb and the lower limb, like I said, they start forming quite early in development. So week 4 and 5 are crucial for limb development. For face development, the major defect is cleft lip and palate, which is a very common craniofacial developmental defect, which occurs. So basically, as I said, in week 7, and week 8, you see the fusion of the different facial prominences occurring. And if this does not occur, then the kid is going to have a cleft. So this occurs during week 5 and week 6. This is the cleft lip, and the palate closes week 7 and week 8. Anything going wrong between week 4 and week 8 can lead to deafness or malformation of the ears. And in the fetal period, which is week 9 to week 38, the ears are little less sensitive to defects. Anything going wrong from week 4 to week 8 can lead to eye defects. Anything going wrong with week 6 and 8 can lead to dental defects like enamel hypoplasia or staining. So basically, you might have teeth that do not have the external enamel. Anything from, like I said before, from 7 to 8 weeks, the palate is most sensitive. So if there is any exposure or any genetic condition that affects the week 7 and 8, it might lead to cleft palate, which is again a very common developmental defect that is seen in kids. And from week 7 to week 9 is where the genitalia is quite sensitive. And from 9 to, so in the fetal period, most of the things are okay. But in the embryonic period, which is week 1 to 8, is when the embryo is very, very sensitive to congenital developmental defects. So this table basically summarizes why the first 8 weeks are important. So as I said, the cleft. So this is a kid with a cleft. These two are kids with developmental defects affecting the face. So in this syndrome, what has happened is that, like I said, there are these population of cells that occupy the developing facial prominences. And if these cells do not give rise to the bone, or if there is some defect in the migration of the cells, then defects like these can occur. This kid here has neural tube defect. So basically, there part of the brain is missing over here. This is again a kid with extremely high number of hairs. So this is a defect in the ectoderm. So the ectoderm is defective in development. So some of these defects are definitely occurring because of disturbance during the embryonic period of development. So moving on, so now we have gone through the first 8 weeks of development, the embryonic period, and I have told you that multiple times that anything going wrong leads to developmental defects. But why do these birth defects occur? So there are multiple factors that can lead to birth defects. It could be a genetic defect that is passed on from the parents to the child, a chromosomal defect, which is where the number of chromosomes are either more or less, or there are structural defects in the chromosomes. And this is again passed on from generation to generation. It could be because of environmental factors, which could be malnutrition, alcohol, like alcohol during pregnancy, tobacco during pregnancy, the mom being exposed to radiation, drugs, or the mom having certain disease during pregnancy. Another factor could be multifactorial, which is a combination of genetic and environmental factors, which is the category under which cleft lip and palate falls, because it has been known to be a disease or a developmental defect caused because of gene environment interaction. And 60% of the birth defects have unknown causes. So this is where clinicians and scientists come into play, where scientists and clinicians are trying to collaborate and trying to understand the pathogenesis of diseases. So there are genome-wide association studies that are done at very large scales in the US, where they're trying to understand what are the causes. Because if you don't know the cause, you cannot treat it. So that's the starter. So here is the same kind of table with a little bit of more information. So moving on to, I have included a little bit of information on human diseases. So I've included one example of each. So the first reason why a developmental defect occurs is genetics. So basically, a gene mutation or genetic mutation carried on from a parent to the child. So this is a child who has a syndrome called as Treacher-Collins syndromes, which is again not a very common, but a lot of cases are seen. And this is caused because of mutation in these 4 genes. So I'm not going to go into the details of these genes, but 40% of these patients have a family history. But 60% of these mutations are de novo mutations, meaning that the parent does not have a history, but the child still has a genetic disease. Why that occurs, we do not know, and it is impossible to understand how a genetic defect occurs, but it occurs at, again, the early stages, which is when the egg and the sperms are uniting. So it's the first week of development. The second type, or the second category, is the chromosomal defect. So Chromosomes are what carry our DNA, and this is a very common syndrome that we have seen multiple cases. You guys might have seen multiple cases of kids like these who have Down syndrome, or also called as trisomy 21. So in this, what happens is that there is either a part or a complete chromosome 21, extra copy of chromosome 21 is present, leading to these developmental defects in the kids. The third category is multifactorial. Like I said, cleft lip and palate affects is a very, very common birth defect. It affects 1 in 700 live births worldwide. And what happens is the non-fusion of the facial prominences, because of which you see these clefts. And there is no cure for cleft lip and cleft palates. The only way these kids are treated is by surgery. So you see the same kid who has been treated, and now he has a nice looking little face. The last category that we know of is environmental, where, like I said, it could be because of exposure of the mother to multiple factors, alcohol being a very common cause. And a lot of cases of fetal alcohol syndrome have been seen, and the kids look something like this. So basically, they have these very typical looking small eye openings, and they have facial developmental defects. If it is a high exposure of alcohol, the kids have something called as fetal alcohol syndrome, where they also have defects in the ear. They have also defects in the fingers, or the digits, also called as clinodactyly. So going through the embryonic development, going through what happens when there are developmental defects, what we do as a scientist is try and understand how can we address this. So you have a genetic disease, but how do you understand what is going wrong in the baby or in the humans, and how can we treat it? So that is what as researchers we are trying to address. And of course, we cannot perform any studies on humans. So researchers use different model organisms for studying development. Here you can see embryos of different model organisms. So this is a fish, this is a reptile, this is a chick, and this is human. So if you see these 4 embryos, you will not be able to tell the difference between the species. And that is the reason why model organisms are used to understand developmental biology and also biology in general, because of a high conservation. Especially during phase development, you will not be able to tell difference in the early stages of development. Along with this, model organisms allow us to understand how genes function, because multiple model organisms have similar or highly conserved genes and genomes, and therefore we can understand the relationship between genes and how they lead to disease, and what are the products that they code for. So what are the proteins that they make, and what do these proteins do? So because of the high conservation, model organisms are ideal systems to understand biology or pathogenesis of diseases. Model organisms also allow us to manipulate the genome. So what I do in the lab is I try to manipulate certain genes and try to understand what happens if you don't have this gene, what happens if you have 50% of this gene. So how would it affect development? And if we get some phenotype, we try to understand what are the underlying mechanisms that contribute to the phenotypes. So model organisms have been used for multiple years to understand genetic diseases and pathogenesis of these diseases. Also, as I said before, development is highly conserved, and therefore they are easy to work with, to understand information about how humans develop. And also, model organisms have been used to study evolutionary biology, so evo devo development. So in my previous lab, we were using the leopard gecko as a model organism, and it is a very cool model organism because leopard geckos replace their teeth infinite number of times. So it was a great organism to understand how we can use the gecko to um, uh, to understand the stemness, or what allows it to develop teeth multiple number of times, and could we use that to apply it to humans because we have only two sets of teeth. So something like that. So model organisms are very cool systems to understand. So we use model organisms to understand pathogenesis of diseases, and eventually we aim to develop cure for human diseases. But it
Pardon,
is not that easy.
ma'am, would
Hello?
you like to take some questions in between?
I am actually on my last slide. So we
Okay.
can
Okay.
definitely discuss. I think I'm on my last or
Okay.
second last. So um, yes, we have a disease. We have resources and funding to understand the biology of diseases. So that is what we do in the lab. We test different genetic variants. We try to understand the pathogenesis of diseases. We try to understand what are the functional impacts of these variants. But what is the main aim of this research is try to understand how can we apply this to humans. But it is not as simple as we think. Like, I get asked these questions like, oh, so how does this translate to humans? And the answer is on this slide. So for discovery of drug, it starts with identification of target, right? And eventually you work for like years and years and years to understand the pathogenesis of diseases, so it leads to some kind of discovery, right? So this, in this, you're spending this much amount of money approximately. You're spending these many years, and then you find some kind of a lead, or some kind of a target where you can target your therapy. So this lead is then picked up and then put into preclinical testing. Preclinical testing is again done on model organisms first before you go into phase 1, phase 2, phase 3 trials on humans. So again, this takes about 6 to 12 years. Then it goes into the phase trials, and then it goes for FDA approval and then post approval. So basically, all of this takes about 20 years, an average of 8 to 20 years, I would say, from discovery of a variant to getting a drug approved. So it's a long process that the scientists have to go through to make something that would really translate to humans. And I think I'm done. I hope that you guys enjoyed the presentation and kind of got to learn a little bit about how we develop and why development is important. So this was a very basic introduction, and I would really like to talk to you all about what I do in the lab, but I wanted this as a very basic presentation where you understand what development is. So in my next presentation, probably in March, I would be talking about how and what we do in the lab to understand these diseases, and how we do experiments in the lab to formulate or to identify targets that would be someday we will be able to translate to humans. So with this, I think I'm gonna stop, and yeah, I can take as many questions as you guys have. Thank you.
So, ma'am, there are some questions in the chat. I'll ask them to us.
Yeah. So I'm gonna. So I should accept.
These will be related to your presentation, and then after that, we'll have some general questions.
Is it just a sec?
Yeah.
Should I stop sharing?
Yeah, you can.
Oh.
But if they ask something, you may need to open it again.
Yeah, I can do that because I'm not able to use both the
Right.
things. I cannot see you when I'm presenting, so it's kind of weird.
Yeah. Athavan, you had a question. Do you want to ask about his question? Okay, I'll read it. So his question was, why do humans don't develop a tail?
Sorry?
Why do humans not develop a tail?
A tail.
Yeah.
So the reason is, so this again goes back to basic biology. So why we do not develop a tail is because we do not have the signaling molecules that would give rise to that appendage at the end. Likewise, we do not develop a beak, or so this is basically just evolution, I would say, is a good answer to this. We would have tails, but eventually we lost them because we didn't need them. So this is applies to all the organisms where eventually if you don't need a certain, so basically that applies to like third molars. We don't need our third molars, so eventually now the newer generations, they're losing them. So it's something like that. And speaking about biology, this is something to do with signaling pathways that lead to formation of certain structures in the body. Like if you induce the formation of beak in humans, and which we cannot do, definitely, but if you induce the formation of a tail or a beak in a human embryo, it would form a beak if you have the exact molecules that would form a beak in a chick or a beak in a bird. So we have done those kind of studies in our lab. We have models where we have induced formation of a lower jaw in formation of an upper jaw in a lower jaw. So you have a chicken with an upper jaw and a lower jaw, and then an upper jaw just here. So you can do that, but we don't need a tail. So yeah, so all of this comes down to a very basic biology with signaling molecules, signaling pathways, proteins, genes, RNAs, DNAs, so all of that.
All right. So basically, a Spider-Man we can make a Spider-Man.
Hypothetically, we don't want to do that, but you cannot do that. That's crazy.
Yeah. Hariharan, you may ask your question. If you want me to read it, you can just write it in the chat.
It's fine, Vansh. I just wanted to know, like, when do, let's say, chickens and humans diverge well in terms of development, that you can't carry your research findings onto humans anymore?
Yeah, so in humans, I think we take a turn around 4 weeks, which is when we start to diverge. In case of chickens, the developmental stages are quite different. So chickens develop in 41 days. So they hatch out and they come out of the egg. But development of the chickens is divided into these different stages, which are called as Hamilton hamburger stages. So in case of the chickens, I would say the developmental period diverges around 5 days of development. they're quite developed by about day 5. You can see all the identifiable features, but about day 7 is when the beak will start to form. So you see a little bit of the beak poking out around day 6, I would say 6.5. So I'd say 5 is a day when it will start to diverge. So it's different in different organisms. So mouse is different because mouse has embryonic days. I would say in mouse, it develops a snout, right? Which we don't. So in case of the mouse, I would say about 12, embryonic day 12 would be a day when it will start to diverge. But I have to say this, which I have been saying for years and years during my PhD. There is a very big debate in the field of research on use of model organisms and why one model organism is better than the other, right? And we always make a case for the chick because the chick is very very close to the humans compared to any other model organism, because in the chick, we agree that it forms a beak, but it before it forms the beak, it's quite similar to the humans, which is, I think, I'm diverging from your question, but something that you guys might be interested to understand, that chick is much closer to human development compared to the mouse.
Yes, thank you very much. And is the beak like cartilage that ossifies, or what is it like?
Yeah, so in the beak, there is a persistent cartilage that stays there forever, but the rest of it is bone. So it
Okay.
is completely ossified by around 12 days after, um, I mean, 12 days of development. It's completely ossified.
Thank you very much.
Yeah.
Aparna, you may ask a question if you're there.
Yeah, I'm there. So, thank you, ma'am. Ma'am, the question I had is that at week 7, then can we have gene editing kind of thing, or is there some kind of, inventions for gene manipulation so that we can prevent all those craniofacial disorders that happens due to the gene mutations, like the neural tube defects and the mental retardation you mentioned?
Yeah, so that's a very good question. Something that is very translational. So, yeah, there are gene therapies that are used to address or treat some of the early developmental defects. But for that, as I said, the scientists have to go through this big whole pipeline to identify a target. In case of neural tube defects, what has been working for years now is folic acid treatment. So, folic acid is something that prevents neural tube defects and has been shown to be a proven drug um, to prevent neural tube defects from occurring. But the thing about folic acid is that it is very essential for the mother to take the folic acid before and when she's pregnant. And if it's not taken, then it might lead to neural tube defects. And the problem with these things is that, yeah, you can develop all these therapies and whatnot to treat developmental defects, but it's it's for the privileged, right? It's not for everybody because these therapies are very expensive, not really accessible to everybody. So the aim here is to do something preventative, right? But in case of genetics, like genetic disorders, you cannot prevent them. But neural tube defects are something that can be prevented, but that requires awareness. So that is something um, that has been used. But in terms of gene therapies, yes, there are certain drugs that are approved for certain diseases. But as far as I know, there are no real treatments for very common syndromes like Down syndrome or NTDs, or atrial septal defects, or those kind of things. Even cleft lip and palate has been like a very, very hot topic in craniofacial development, and there are multiple GWAS studies that are done, but nobody knows how to treat them. And there are like multiple labs, there's like millions and millions of dollars being spent to understand the pathogenesis of cleft lip and palate, but it's hard to pin down something, because when you have so many genes contributing along with environmental factors, so this is all like personalized medicine, right? So yeah, so far there are gene therapies for, I would say, cancer and stuff, but not a lot for developmental diseases.
Got it, ma'am. Thank you, ma'am.
Mhm.
There's a little different kind of question from Sivaranjani. Please ask.
Hi, ma'am. Actually, thank you for the nice informative presentation. It's very useful for us. And as you said, the most 60 percentage, the reason may be unknown. So, if we saw someone who is suffering from this disorders, how can we help them to get the right medications that is available in India, or especially that economically poor people? How can we help them?
Yeah, so that's a good question, and I would say a good approach as well.
Thank you.
Um, so if you see a person with a disorder or something that you see is abnormal, right? The first thing is to do a genetic panel, right? To do a genetic panel to understand what are the gene defect. Does this person have any genetic defects? Secondly, get a family history. If the parents are willing to contribute, do a genetic panel for the parents to understand if it is an inherited genetic disorder. So you have to cover all the three things, right? You have to cover if it is a genetic disorder, if it is a chromosomal disorder, or if it is an environmental factor, right? You have to cover these three things. And in order to cover these three things, you need to do a genetic panel, a chromosomal panel, and get a family history to understand if the child was exposed to some kind of teratogens or some harmful drugs or anything like that when the mom was pregnant. After this, if you find something, there is nothing like a treatment for developmental disorders. Once you have it, you have it, right? You cannot correct them. So for example, for cleft, you can do a surgery and correct it. If you are dwarf, you cannot do anything about it, right? You cannot increase the height of a person. I mean, people have claimed that you can do that using what was some kind of like stem cell therapies, but I personally do not believe in that. But those kind of things. But this is all palliative treatment, right? This is all you trying to help the person as best as you can to live a normal life. So I mean, the approach is really great, but I think um, in terms of helping them, the only thing you can do is just do stuff that can make their life easier, because there are no medications. So, for example, there are diseases that affect the bones, right? If the bones are not ossified, the bones are not forming properly, yes, you can do therapies for those. But in terms of disorders that I discussed in this presentation, there's definitely no treatment for those, unfortunately.
Okay, ma'am. Like actually, I have a cousin, one from my dad's side and one from my mom side. One person is deaf and one person is have that palate cleft. Like they had, she had surgery in her childhood. Now she is okay
Yeah.
and leading a normal life. That kind of thing is manageable, quite manageable. But the thing you have shown in the picture was certain condition were really worse to have a normal. So comparing with that, she is okay now, and one
Yeah.
person is deaf, no, so that we can't do anything.
Yeah.
So like that, I'm just curious to know that reason and treating them.
Yeah, that's I mean, that's unfortunate that you have this in your family.
Yeah,
But I have to say that with clefts, it's not, the surgery is not the end all cure all kind of thing, because these kids have a lot of other problems. They have speech defects, they have dental
correct.
defects, they have like their jaw doesn't grow properly, so their upper jaw is smaller than the lower jaw. And
Mm.
also the I mean, I have seen a lot of these cases when I was practicing as a dentist. And the main thing I also feel about these patients is that they lose their self-esteem, they lose the
Correct.
confidence, and that is something that is very it's kind of sad. But yeah, so they need support throughout their life. So it's not
Yeah.
just you did the surgery and that's the end of it, right?
Mm.
So yeah, I would suggest, being mentally there, emotionally present for a person who's suffering from some of these diseases.
Yeah, correct.
Yeah.
Thank you, ma'am.
Hmm.
We have two more people who have questions. So, Abhishek, you can ask a question. Are you there, Abhishek? Okay, I'll come back to you. Then, Kush, you can continue with your question. Kush? Mm, okay, so I'll read your question. So I'll take both of these questions. Abhishek asks that how do genetic mutations in specific genes contribute to the development of common diseases like cancer or diabetes? And uh, can genetic defects be recognized and cured with the help of recombinant DNA tech? Both the questions.
Yeah, so addressing the first question, yeah, cancer and diabetes are the hot topics in research. And um I would say, yeah, unfortunately, they are common diseases. And yeah, like in case of cancer, there are multiple genes that are identified that lead to various types of cancers. In case of diabetes, it's specifically the islet cells, which are the cells in your pancreas that are defective. And there are certain genes that ISL1, which leads to diabetes. But the problem, I mean, with these genetic mutations, you feel that, oh, now these genetic mutations have been identified, and oh, the scientists should have a cure for cancer, right? I mean, that's what I hear from a lot of people. Oh, so you're gonna cure cancer, and it's like, no, dude. It's not that easy, because there's so many genes, there's so many different types of mutations. So basically, one concept that I can explain to you guys if that's okay. So we have a DNA. All of us have a DNA, right? The DNA is then converted into RNA, which is again a very important molecule. The RNA is then converted into protein. This is like a normal pipeline, right? In anything functioning in your body, the cells are talking to each other, the cells are dividing, everything is happening in the background, right? This is all autonomous. So DNA converting into RNA, this is called as translation, transcription, translation. So with genetic mutations, what happens is that there is a little defect in your DNA, right? It could be a small defect, it could be a big defect, but this defines what kind of RNA it forms and what kind of protein it forms. Sometimes it does not even form RNA, sometimes it does not even form protein. So every cancer has multiple genes, and multiple genes having different mutations at different points along the DNA. And this defines what kind of protein it forms. If it forms a shorter protein, it forms a longer protein, it forms no protein. And this defines how the cell functions. So it is much, much, much complicated than just knowing the genetic mutation, because what we do in the lab is we test these proteins, right? We test what happens when you have these defective proteins in a model organism. So yes, understanding specific mutations is something, but yeah, understanding how they function is takes a really long time. And addressing the second question about recombed DNA, it has been used recently in the labs a lot. We use, I mean, it's basically artificial DNA, right? Recomb means anything that is artificial. So you introduce this artificial DNA and you get it to work, get it to form RNA, and get it to form protein. So it has been used for certain cancers, but it's still evolving, I have to say. So I wish it will evolve and help people, but yeah, uh, does not apply to developmental biology again. And is that it? That those were the two questions, right? Oh, I can hear you.
Your mic is off.
I'm sorry, I'm sorry. So Kush has another question. Has someone in history tried to do something crazy with human embryo, like inserting other animal genes in human embryo? And I have a follow-up question to that. That is, so like we it's easier to induce diseases in humans than to cure them, right? Like we can easily actually do many things with the DNA.
Yeah. So to answer the first question, human embryos, no. It's it would be crazy to do something like that, but this was like really, like, I wouldn't say very, very long time ago, but people used to do crazy stuff back then when it was okay to work with humans. So use um, human embryos for experimental studies. So I have seen some of those papers, but not really to induce something like induce a mutation and create a monster. No, I don't think scientists are really interested in doing something like that. And your question was, what was your question, Vansh?
Just a simple like it's like easier to induce diseases in any organism than to cure them, right?
Yeah, that is true because we understand the pathogenesis of diseases. Um, so in animal models as well, we can. So basically, what we do is we introduce a mutation, we create a defect, and then we understand what is causing it, and then we also create treatments in animal models. So you will see a lot of rescues being done in the animal models, but again, it may or may not work in the in the humans, right? Yeah, but you're right. Like it's easier to do something destructive, but difficult to, you know, do something constructive, like it is in everything in life.
Uh, yeah, Abhinav, I think this is the last question, and then we have just a general question that was posted before the meet. So Abhinav, we can take your question.
Hello, ma'am.
Mhm.
So, ma'am, I have two questions. My first question is that why the egg is usually fertilized by only one sperm?
Egg is, sorry.
Usually
Why?
fertilized by only one sperm.
Egg is usually fertilized by only one sperm because I mean the egg can only take one thing at a time. So like in vitro fertilization, I don't know if you guys have like really watched those videos. You can have multiple sperms attacking the egg, but only one penetrates, and when one penetrates, that's done deal. You cannot have too many um, sperms, you know, getting inside. So once it enters, that is it. So that's biology.
And ma'am, I have a second question. It's a crazy question. Maybe you laugh. So, can I ask?
Yeah, sure,
Okay.
go ahead.
Yeah. So, ma'am, just like, can we send human sperm in animals' embryos to born a human like babies?
I mean, you can do that, but it's I don't think it's gonna do anything. I mean, in mythological studies, yes, you know, um, animals were used to like make human babies, but I don't think it works that way. But to be honest, I haven't um, read about anything like that, and I don't know if it can really happen. I'm sorry, I cannot answer that question.
Okay,
Okay, ma'am.
there
Thank
was
you.
a really hyped up question. So it's not related to exactly this webinar. They asked that they wanted to know how can they link this data science and all to neuroscience or biotechnology or anything related stuff, and also like are there any kind of future earnings or research opportunities?
How can you apply this webinar
No,
to
no, no.
biotech?
This web it is not related to this webinar. They're asking
Oh,
about like data science and all. How can it be connected to biology, basically?
oh, so right now is the best period for biotech, bioinformatics, data science people to dive into biology because right now is the time for big data in biology. We are doing a lot of sequencing studies, and that requires a huge knowledge of bioinformatics. So something that you guys do probably, which is Python, R, you know, those kind of coding things. Um, you can definitely apply that for biology because what we need is to use these sequencing data. So sequencing means we get a certain tissue from the animals, and then we make a RNA profile, and then we get a huge list of genes. So we use this gene to identify targets, like I said before. So this requires coders like you guys to help us with simplifying this data for us. So identifying what matters and what doesn't matter. So that is how data science people can um, get engaged with biology.
I hope it answers all of your questions. So we have a surprise. Vikas, you may continue.
Hello, am I audible?
Yeah, yeah.
Yep. Hello, guys. I hope you enjoyed this webinar. It was a really different webinar and topic that we usually do. Data science people, coding one. We are usually into coding stuff, internships, data science thing. So it was really a different thing. You should know that how the human thing like we develop, it's really important. So I hope you enjoyed. Thank you, Dr. Shruti, for coming and giving us the presentation and explaining and taking doubts. So yep, here on behalf of Dr. Shruti, we have a small surprise. Just give me a second. um yep, is it visible? Yep. So just, ma'am, can you tell about this book? uh, you are muted, ma'am. You're you're muted, ma'am.
Oh yeah, sorry.
Okay.
Yeah, so this is Scott Gilbert. So this is like a very interesting book for people who are interested in developmental biology. It will walk you through much more details about how we develop, and also I think it will give you information about model organisms. It will give you information about how basic development occurs in different model organisms and how these model organisms can be used to study development. So it's a very interesting book, but it depends on how interested you are in developmental biology. But yeah, this is something that
Yep.
that is a good starter for people who are interested.
So
Yeah.
people who are interested and want to learn biology, developmental biology, so they can just fill the form, and we'll also give you a form in your like mail. So just please fill it, and we'll definitely provide the ebook and the PDF to you. So I hope you will read it and give some time, not just reading 1 2 pages introduction. That's all. Seeing images. No, please give us some time. 10 minutes day will be fine. And like second thing is the next thing that we are going to do is the workshop on front end development. So Abhinav, sorry, Agrim, are you here?
Yeah, before
Aviral.
that, let me just.
Okay, so
Yeah, it
great.
was actually nice to see data science people to be interested in this, and we could have a follow up. Like if we guys are interested, so we'll post a form. If you guys are interested and all, you can just fill it, and we'll see if we can keep another meet with Dr. Shruti, where she can tell about her research. And thank you so much, ma'am. It was really nice. Actually, we actually all enjoyed, and it was like really good. Thank you, ma'am. You can continue,
Thank you
Agri.
for having me. Great.
Yep. Viral, can you just give the description or just a small gist about the workshop, that what we are going to do in the workshop? It's Viral, are you here?
Yes, I'm here.
Okay, please.
Hello, am I audible?
Yep.
Okay. Okay, so this is a workshop regarding the front-end development of website development, if you have ever heard about it. So what we are going to do is that we will be introducing you to HTML, CSS, and JavaScript one by one. First, we will go through the HTML part on the 26th and 27th. The timings will be from 7 to 9, 2-hour session, and we will also be providing you with minor assignments and major assignments regarding that. After that, we will be moving to the CSS parts, And again, it will be a 2 day workshop from 30 to 31st. Okay. And the timing would be the same from 7 to 9 p.m. And we'll be again providing you with minor assignments and major assignments regarding this part 2. And same goes for the JavaScript, which will be from 4 to 6. In JavaScript, we will also be introducing a little bit about Node.js. We'll not go into deep knowledge of Angular, Vue, React, etc. We'll just be doing the basic things of JavaScript, like how do you code and how the Node.js works. And then there will be a final project. Will be accumulating the entirety of the knowledge that we gain from HTML, CS, and JavaScript through this entire workshop, and we'll be creating a project so that in future you could create your own websites and get started with your front-end development journey. Okay, thank you.
Okay, thank you,
Oh,
Viral.
so if you guys enjoyed the webinar, we'll be posting a feedback form just in a while, and please fill it. Please give your reviews and everything. If you
No,
guys have any questions, I think the feedback form, yeah. So if you guys have any questions from Dr. Shruti or in general biology or anything, just post it.
and
Thank you
yep.
so much for
For
joining.
that,
Yeah,
uh, I think you can just take a screenshot of this roadmap. It will be like helpful for you. And yep, if you have any query or if you want to like know about us, anything, any suggestion, just please mail it as community@stemonef.org. I will definitely answer you. Yep, Gaurav, this is the surprise, or
yeah.
what you were expecting.
The book and the
The book is the surprise, and
yeah.
the workshop is the surprise. And
Mhm.
yep, there will be many surprises. Just please join our WhatsApp community and on LinkedIn. So the recent updates and the updates which will be coming, you can just check them. And yep, thank you for joining. And yep, Dr. Shruti, thank you. Like you joined, and
Thank you for giving your time, ma'am.
You're welcome. I
yep,
enjoyed it. I hope everybody did.
Yep.
Yes, ma'am, definitely enjoyed.
That's
yep,
great.
and Pankaj, and if Pankaj, Siraj, anyone is here. My, Mayank, are you there? Sagar, Pankaj,
What do you need?
just want to ask them, should we end this thing, this meet, or
Can I drop or
yes, ma'am,
you need
sure.
me
Like,
on?
uh,
Okay.
I think, yeah, ma'am. Thank you, ma'am,
Thank you,
for
ma'am. Thank you so much
Awesome.
for your time.
Thank you.
Thank you, ma'am.
Have a good night, guys.
Good night, ma'am. Thank you.
So thank you, ma'am, for being here.
Mm-hmm.
Things are done.
yep,
Awesome.
Yeah,
Pankaj,
because yeah, I'm back. Yeah,
can we stop, or like, can we
yeah,
stop?
sure. Let's wrap up.
Okay. Yep. Thank you guys for joining. Really,
So,
really, we
hmm.
appreciate your participation. Just join our community if you want to see more things related to STEM, workshop, webinars, project. Please do join our community. Yep. Thank you.